Okay. Little uh, housekeeping here. Okay. It's Jay Herson. My talk, Bias and Randomization, 1980, 2020, 2060, was presented on February 11th, 2021 in an IDDI webinar. This all began in 1980. I wrote a paper, Patient Registration in a Cooperative Oncology Group. It appeared in the first issue of the new journal, Controlled Clinical Trials. And I came across this paper in March of 2019 by accident. And I visited Brussels right after I discovered it, and my good friend Mark Boyce, the founder and CEO of IDDI, I'd asked him, I found this paper, would it be good for me to write another paper? This is the way we did it in 1980, this is the way we do it today. And Mark said, no, not a paper. We can have a full-day symposium, and we can get sponsorship and hold it at the Magritte Museum in Brussels. And we'll have it in April of 2020, the exact 40th anniversary of the publication of your paper. And I was somewhat skeptical, but a few months later, lo and behold, they secured everything. One detail they could not control, and that is in April of 2020, there was the pandemic. So we had to cancel the meeting, but I am delighted that they invited me to give this talk, which was to be the keynote talk of that symposium, as a webinar. And as a keynote talk, in my opinion, my job was to tell the participants, the audience, what they should be thinking about as they hear all of the other papers during the day. But I think this will do well as a standalone paper as well. So as an overview, we're really talking about persuasive evidence, which I will define. And then the challenges to randomization in the 1970s and historical controls, how we did things in 1980. In 2020, there's been a rethinking of much methodology. And in 2020, we have historical controls to deal with again. And also real-world data. And on our way to 2060, we will stop in 2025 for me to explain some things that I see in the near horizon. Then we'll arrive in 2060, but we will be talking about persuasive evidence throughout. My first hero is Jerome Kornfield. Kornfield was one of the founders of biostatistics at the NIH in the late 1940s. He became head of biostatistics at what we called the National Heart Institute at the time. And then after retirement, he was faculty at University of Pittsburgh and George Washington University and founded the Biostatistics Center at George Washington University. At a conference in 1973, I heard him say, clinical trials are cogent description. And I thought that was very clever, especially when I looked up what does cogent mean, and cogent means persuasive. And a clinical trial must be persuasive. It must yield persuasive evidence. And again, that's what we're going to be talking about today. So I will use the terms cogent description and persuasive evidence interchangeably. There were challenges to our belief in randomization in the 1970s. Ed Guillen and J. Freireich in 1974, C.D. Hogginson, 73, and Bernard Fisher in 1973. The Guillen-Freyreich paper appeared in the New England Journal of Medicine in 1974. And J. Freyreich passed away on February 1st at the age of 93. So I'll try to be kind to him. But they wrote a paper, both at MD Anderson, and Ed Guillen eventually became my boss at MD Anderson. They wrote a paper saying it is unethical in most cancer clinical trials to have either a placebo control group or an active control group with an already approved drug, because those drugs were known not to be effective. And what would be better is to do a one-arm trial and compare to data that are in the literature, or perhaps a control group could be found in a previous clinical trial. In other words, no randomization to a concurrent control group. Created a lot of controversy when this came out. Now, in the field of breast cancer, Haagensen was in favor of the radical mastectomy, and he felt in 1973, as people were beginning to doubt that every woman should get a radical mastectomy. And he said there's this crazy guy in Pittsburgh, surgeon, named Bernard Fisher. Not only is he advocating what they called a simple mastectomy back then, today we might call it a lumpectomy. He's not only advocating that, he's randomizing patients in a clinical trial. So chance alone will determine whether a woman gets this inadequate, in my opinion, simple mastectomy or a radical. And this stirred up also some controversy, because there was already controversy about the radical mastectomy at the time. Now, 1980, clinical trials in cancer was organized in cooperative oncology groups, and still is today. But let me indicate that although I'm going to be mentioning oncology, what I say today applies to all therapeutic areas. There was SWOG, the Southwest Oncology Group, that had the statistical office at MD Anderson in Houston, and that is where I worked starting in 1976. There were others. The EORTC was the European cooperative group, still exists today, and the other groups on this list still exist today, although their names have changed, or at least slightly. All trials back then were open label, and the data were continuously analyzed. The results were displayed by treatment group and the p-values at regular meetings. Yes, there was type 1 error inflation, but we didn't talk about those things in 1980. So I wrote this paper in 1980 after I had left working for SWOG. But when I worked for SWOG, 1976 to 79, I was in charge of the biostatistics for all of the pediatric oncology trials. But at my first meeting of SWOG, my first group meeting, 1976, all hell broke out among investigators, claiming that they were not given credit for all of the patients they registered. So we came back to Houston, and I worked with a computer programmer, and we traced them, and in fact, there were no errors for that. It was just a timing issue. However, in looking into this, I saw many other problems with the way registration was being done. So I volunteered to overlook registration and randomization for all of the trials in SWOG. Now, in 1980, the drug candidates were all owned by the NCI, and the cooperative groups wrote protocols to use them. The drugs were manufactured under an NCI contract. There was little presence of pharma companies or the FDA at these meetings. Most drugs were under an IND, pre-market clearance, in order to begin a clinical trial. A few were approved and marketed by pharma companies. Now, those that were approved, you see them in the last bullet here. These drugs are still in use today, but not as the primary drugs. Phase 2 and 3 trials were a mix of pre-market and post-market drugs. There was no site monitoring, as is common in the pharmaceutical industry today, or drug accountability, also common in the pharmaceutical industry today. There were frustrations in the cooperative groups. Investigators were not happy with the NCI drug inventory. They were hesitant to use drugs that had no obvious rationale. They tended then to write protocols with only slight differences from recent trials by other drugs, where at least they knew something about the safety of those drugs from these other trials. Then the groups were put on probation or defunded for not having innovative ideas. It was a catch-22. How did we do patient registration in 1980? Investigator made a telephone call to a registration assistant at the coordinating center. In this conversation, eligibility was established. Randomization was via a pre-printed blocked lists and trial-wide. So there'd be a book, and then there'd be a list, treatment A, treatment B, A, B, and then the new patient would be entered on the next open line. And if there was an imbalance within site, a treatment imbalance, registration assistant would then switch the treatment without telling the physician. There's nothing dishonest here. We're trying to balance the treatment arms within a clinical site, and but to start discussing that with a physician would just open up a Pandora's box. Well, there were some biases in this, not more than just a few. We permitted off-hours randomization because we couldn't have 24/7 coverage. So it was done by a coin toss and the honor system. But we detected an overuse of off-hours randomization, and we kept monitoring that. We knew that oncology patients are not registered at night or on weekends, but it would happen occasionally, but not as often as we had seen. Then there was a wait and see bias for both single arms and even in randomized controlled trials. But let me just, for the sake of time, let me just describe what it was on a single arm trial. The physician would start treating the patient. He or she would wait to see how well the patient did. If the patient did well, call the coordinating center and register the patient, single arm trial. If not, we would never hear about that patient having been started and stopped. There was no drug accountability then. The investigators would say, send me another dozen. There was no one who could say, wait a minute, you only registered 3 patients. There was none of that. Many ineligible patients were randomized, and we considered them non-evaluable in our analysis. So a lesson learned here, randomization alone does not imply cogent description. There was an anti-randomization rhetoric. Statisticians were a deep state conspiracy against physician judgment. Dial a phone number, and a clerk tells the physician how to treat the patient. That second bullet was a ridiculous misstatement. My second hero is Tom Chalmers. Randomization from the first patient, he said over and over again. Tom Chalmers was one time in his career, the director of the prestigious clinical center at the NIH. He went on to become the dean of the Mount Sinai School of Medicine. After his retirement, he was involved with Harvard and the VA in Boston in many activities. The founder of meta-analysis, I believe the first president of the Society for Clinical Trials. He told me a story once at a meeting that he was a physician in the Army during the Korean War. And you remember the American TV series MASH, doctors doing surgery under the tents not far from the front line. Well, he was an internist, not a surgeon. But in one of those MASH tents, he was randomizing patients to two different ways of treating hepatitis in the battlefield, while he could hear gunfire in the distance. This was a man who believed in randomization. When he was dean of the Mount Sinai School of Medicine, I would not have advised a faculty member to go into his office on a Friday afternoon with a grant proposal that calls for a non-randomized study. Just saying. We had one scandal before cable news in SWOG, a leukemia AML trial, 1978. Leukemia trials, as some of you know, have a common induction arm. Then when we get to the maintenance phase, that's when we randomize to two different ways of maintaining. And treatment A might be W plus X, where W is a standard treatment, but possibly not FDA approved. Today it would be, but back then the standard treatment didn't have to be FDA approved. But X is the experimental treatment, and that was possibly already FDA approved, different from today. Nevertheless, we called it an add-on trial, trying to find the effect of X, and we still call it an add-on trial today. Well, what happened? In preparing for a SWOG meeting, I looked over all of the trials that were to be presented, and I find a phase 3 trial with, it looks like, close to 2-to-1 randomization between the experimental and the control arm. And I said to myself, I'm not aware of any active phase 3 trials that have 2-to-1 randomization. So I went back and looked at a lot of files, all on paper in those days. And I find out there was an agreement with the coordinating center before I got involved that Dr. Z from MD Anderson would secretly arranged for all of his patients to be put on treatment A, the experimental arm. MD Anderson was the highest enroller. That explains the 2-to-1 randomization. It was later discovered that Dr. Z followed his own induction treatment, not the one in the protocol that everyone else followed, regardless of treatment group. Dr. Z used the resources of SWOG to test his own hypothesis and provide the better treatment for his patients. In 2012, these two talented authors wrote the book Cancer on Trial. It was the history of clinical trials in cancer. And in it, I read that in the 1970s, the era that I'm talking about, there were many arguments among the physicians in the NCI, cancer treatment branch, that were arguing, should we be allowing randomization? Is it ethical? Isn't it? And they argued back and forth. I'm sure there were no biostatisticians in any of those meetings. Interestingly, while I was at SWOG, some people about my age were fellows. They were young physicians, fellows in oncology at the NIH. And one of their jobs was to go around and attend the different cooperative group meetings. And one fellow I was friendly with, I told him, look, there are all of these problems in registration and randomization and blah, blah, blah. And this trial, this AML trial, we had these problems. And he said to me, I don't quite understand what you're talking about, but I think you're saying you don't like your job. But no one at the NCI cares if you don't like your job. So I stopped talking to him. He was going to be no help. But interestingly enough, he was the only one in that fellow class that made a 40-year career as a bureaucrat at the NCI without treating ever treating another patient. Just saying that, I don't know what the significance of it is. Come 2010, the NCI approaches the Institute of Medicine and they say, there's problems with these cooperative groups. Someone's got to look into this. Will you look into it? Look at how many years later this was, 30 years after 1980. And so they did, and they say the RCT is the gold standard. They even advocated minimization. Well, what do we do in 2020? We have double-blind trials. Randomization is web-based, sometimes endpoint adaptive. We do an unblinded analysis only at the end of the trial. But the pharma industry owns all of the new drugs from day one, not the NCI. So good clinical practices have come out from regulators, from the ICH, the International Council on Harmonization, and these are more powerful than the NCI because the pharma industry wants to get their drugs approved and they must go through good clinical practices, and in most cases they agree with the GCPs. In 2020, the cooperative agreements replaced the grants that we had for the groups. So in the cooperative agreements, the NCI has input to the protocols along with the investigators of the group to prevent someone from going in the wrong direction. Phase 3 trials are run by multinational CROs. Sometimes a cooperative group is a subset of total patients. So today, SWOG might have put 500 patients on a trial, but there might be 1,000 patients, the other patients coming from all other places. Phase 2s sometimes are RCTs today. And so randomization is generally accepted, and it's even used with minimization. But one question we have is who to randomize. Underrepresented in clinical trials, children, seniors, minorities, pregnant women. We have the master protocols. There are many versions of the master protocol, but the one most common is different drugs will cycle through quickly, one at a time, treating patients, and we look to see the ones that have the most promise, all under the same protocol instead of starting a new protocol for every drug. And but there is no randomization in most of those. The SMART trials, we randomize too much. In a SMART trial, we start treating patients, and if a patient has an adverse event, we randomize them there, 2 different ways of treating an adverse event for a patient on this particular drug. If a patient relapses, we randomize patients between 2 ways of what comes next for them. But there are many problems with having too much randomization. There are post-randomization headaches, especially with our current discussions about estimands, intercurrent events. How do we handle them? Rescue meds, missing data. How do we handle this in our analysis? The 1980 paradigm was trial design, write a protocol, randomization, and then an efficacy and safety analysis. Look at what we have to do today in 2020. We declare estimands, we design a trial, write a protocol, write a statistical analysis plan. We didn't have that in 1980. How are we going to handle missing data, the treatment switching policy, real-world evidence, the program safety analysis plan, the find a data monitoring committee, then do randomization, efficacy, safety analysis, and then a sensitivity analysis because of all of the assumptions we made in the bullets before this. In 2020, there's been a rethinking. Intent to treat, or the treatment policy, has been around since the 1970s. We just randomize patients, and whatever happens to them, what we're randomizing patients to is a policy of treating them with treatment A or treatment B, not to what happens to them afterward. But with our interest in estimands today, there are other ways to look at this. And there is a claim that this is a 20th century estimand. The Cox model, the proportional hazards assumption, is under siege. The p-value, whoever thought that would be under siege, although I think that's long overdue. Why does it have to be 0.05, and why is it so important? And literature-based meta-analysis, I never was comfortable with that. Whenever I tried to do this, I couldn't find the data in the same form or even at all in the literature of the previous clinical trials. And randomization is also a rethinking today. We have historical controls again, more sophisticated this time. Bayesian borrowing from historical data for the control arm drug. So we have concurrent controls that are enhanced by historical data. So in other words, we might randomize 2-to-1, twice as many patients on the experimental arm than on the control arm, but we enhance the smaller control arm by Bayesian borrowing, covariate matching from historical data. So randomization is preserved in this form of historical controls. We have non-randomized historical controls, no concurrent control arm at all. This is done usually for the orphan indications where there are so few patients, most of them childhood congenital enzyme deficiencies. Post-market confirmatory trials for an accelerated approval drug, well, it would be hard to randomize patients then, even 2-to-1, because it's already known that the drug has some kind of efficacy. So we do it in these cases. The methodology used in these are the propensity matching on covariates from historical controls, and we can use real-world data to create synthetic controls. Bayesian borrowing, as I said, which is matching and borrowing based on the covariate data. It's doubtful, problems with this is it's doubtful that data on intercurrent events, rescue meds, would be available in synthetic control data. And there's a dilution of AE incidence for AEs dependent on periodic observation. So in other words, in the experimental arm, patients have their ejection fractions measured every 4 weeks. In data that we use from the literature, from claims data, wherever, it's doubtful that it is measured that often, and perhaps only when there are symptoms. And it's hard then to compare with that important measure of cardiovascular toxicity. Now the FDA has had opinions on historical controls that should be used, or they are willing to consider it, in diseases of high and predictable mortality and predictable course. Effect of the drug is self-evident, small population and unmet medical need, and the efficacy is believed to be substantial. Well, if all of that is true, why would we even be doing a clinical trial? I don't doubt this has been done, but very rarely in medical history. Sponsors and historical controls. The sponsors are gaining experience with Bayesian borrowing in their internal analysis as an aid to design an analysis, even though they may not be able to submit to regulators. So that before they submit, they're doing all kinds of sophisticated analyses with control groups, getting data, perhaps historical, in various ways, first for a go, no-go decision, but also what is the best way to analyze the data given what we see here. When it comes time to submit to regulators, it will be a frequentist analysis. Sometimes they simulate historical controls from the literature data for use in this pre-submission research. We have today science via legislation, the 21st Century Cures Act and Prescription Drug User Fee Act. They advocate use of real-world data. Risk-benefit analysis should be done. And innovative designs and analyses, definitely. Of course, it is senators who voted on this, not people who actually have to do this research. It's not all that easy. From the Prescription Drug User Fee Act, I believe one real advantage of that we've had it now since the 90s. But the sponsors of the submissions, drug companies, they pay a fee for the benefit of having their drug reviewed. That, of course, that doesn't guarantee it'll be approved. But with the money that the FDA got from these user fees, they're able to go out and hire people, physicians and statisticians and others, with the same contemporaneous recent training as physicians and statisticians at the FDA. They can talk to one another about these things as they never could before. Real world data, well, Cancer Link is a network of oncologists in practice all over the world. They fill out forms, sending data electronically to a central clearinghouse at ASCO, and a ASCO member can type in the characteristics of their current patient, and they can see how other patients of that type, which might not be represented in clinical trials, are treated. FDA Sentinel project, bringing together data from claims data, a lot of insurance companies. Of course, Europe has the state-run health systems with lots of data. Medicare data, electronic health records, I said insurance claims. And the problem with doing this is the stakeholders, be they drug companies or physicians or whoever, they'll support or reject inference from these sources of data, depending on the results and their own bias. If they get the result they want from a analysis using historical controls, real-world data, then they say, there it is, we've shown it. If they don't get the result they want, they will say, well, only a randomized clinical trial can answer this question. Didn't you hear Jay's webinar? So again, it's going to be difficult to get agreement here, but we're making progress with it. Most of these sources of real-world data yield only safety data, insurance claims, but very soon all sources will have efficacy, intercurrent event, and biomarker data as well. Sophisticated analysis of real-world data, non-randomized, can provide insight to issues like treatment efficacy heterogeneity over subgroups. We really can't do a good job of that with a clinical trial. We don't have enough subgroups represented, enough patients in the subgroups we're interested in, etc. Will this yield persuasive evidence? To whom? There are modeling controversies, of course. The 2020 paradigm includes analyses that utilize models that depend on assumptions that cannot be verified. Missing data and propensity scoring, models here, how we use them, what are the assumptions? Sensitivity analysis, yes, we do it, but it's selective. Why do we see only these sensitivity analyses? However, to deny the implications of data analyzed with these assumptions is to deny our ability to reason. Is our definition of cogent description changing? Let's stop in 2025 to look at the short-term future. Randomization in pragmatic trials, randomization combined with real-world evidence, and centralized statistical monitoring will replace site monitoring. Sending the armies of people out to the clinical sites, going over all those data was not targeted and didn't produce as much as centralized statistical monitoring is already demonstrating it can do. And of course, during COVID, it was used much more than it was before COVID. The clinical trial enterprise, Rob Califf put this forth about a decade ago. Rob was the vice chancellor for health sciences at Duke University, and he was the FDA commissioner in the last year of the Obama administration. And he created this clinical trial enterprise. He put it forward as a proposal. Patients visit their own physician with some medical issue. Physician enters the data into the EHR, and a list of clinical trials this patient is eligible for appears. If the patient consents, their own physician will be an investigator on this trial with at least one patient. This patient might be the only patient they put on, and the patient is, of course, randomized right there. Now, what Rob Califf envisions is that all physicians in their residence program will be trained in how to be an investigator in a clinical trial. The central stratum of this submission, of the data from this, will be patients who satisfy the regulators and sponsors' ideal patient, no comorbidities, no current treatments, other types of treatments. We always see those, but there can be separate strata for elderly patients, patients taking only one drug, patients with only one chronic condition, etc. So we'll get more data. We get more data than just what the regulators want, which would be valuable. Also, this scheme would be attractive to elderly patients because they're going to be treated by their doctor, their same doctor in the same office they're used to going to. Many elderly patients are not eligible for trials, but when they are, they're hesitant to go to a strange doctor's office and even sometimes to travel to another city to be in a trial. So this would get us more elderly patients on the trial. But this CTE has not gained traction. And I tell my students in biostatistics, well, some are physicians also, I say that maybe it's the name. Maybe if he called it the Califf conjecture or Califf's last theorem, it might be more attractive, especially to statisticians. In COVID-19, there are many innovations, and one is decentralization. Some trials recruit by social media, some self-enrollment over the internet, some randomization and shipping by mail pharmacies, some local labs for the blood draws, and outcomes by email, of course, telemedicine and home visits by nurses. Not all of these would apply to every trial, but I think they'll be used more as we have gaining experience during this pandemic. Well, here's a trial of interest, the Zostavax trial for the herpes zoster vaccine. Not the vaccine most of us are interested in today, but an important trial. The sponsor did a RCT, 22,000 subjects, 50 to 59 years old, with a median follow-up of 1.3 years, and they found vaccine efficacy of 70%. They received from this a provisional approval. They made a post-market commitment, an observational study, 1.3 million Kaiser Permanente members with covariate adjustment in the final analysis. So they had more data on the 50 to 59-year-olds, but also the 60 to 69, 70 to 79, and 80 plus. Overall, vaccine efficacy was around 60%, and they got a final approval. This is a marriage of RCT and RWE, which I think we'll see more in the future. Now, with all of this talk about real-world evidence, I should mention that exactly a year ago, these highly prestigious authors wrote a paper in the New England Journal of Medicine, The Magic of Randomization versus the Myth of Real-World Evidence. And they point out different trials where there was both a randomized study and they looked at real-world evidence, and the randomized study had the correct results, and the RWE was misleading, and we must be careful, and that's what they are cautioning here. With the help of Magritte, I wanted to include Magritte, even though I couldn't give this talk at the Magritte Museum, but Magritte is taking us to 2060, the future. Let's assume cancer is not cured. 2060 will have quantum computing in the cloud. Now, does anybody think that computers with this kind of power would be used to compute chi-squares on a 30-patient experiment? No, they're going to be getting data and aggregating it from all over and analyzing it very quickly. Medicine and data science will be one profession, not separate. And there won't be training where, okay, you study medicine Monday, Wednesday, Friday, and data science Tuesday, Thursday. No, it all be incorporated, is all one science. People like myself, a biostatistician, will be similarly trained pharmacology and data science together. That is for a clinical trial statistician. Pharmacology and data science trained together as one profession. Now, however they're trained, they'll all be driving electric cars by then, I'm sure. Machine learning will guide medical practice, choosing drugs and doses for a patient and changing when appropriate. So machine learning will always be with us. The physicians will automatically at every time the data are entered from a patient, machine learning will guide us. But clinical trials will feed this machine. Results of clinical trials, when we didn't have all the answers, will feed this machine. Synthetic biology will be creating friendly life forms, microbes that will go into our body and help cure diseases and help in treatments and prevention and treatments in various ways. Patient evaluation and drug supply at home, capabilities of handling massive data, clinical trials, wearables, clothing, EHR, sensors in the home, prescription data, claims data, and all sensors in the home and in wearables will be passive. We don't have to turn anything on, just walk around and data will come. There'll be no blood draws necessary. The sensors will determine all of those hematology. Machine learning will replace human committees, data monitoring, safety assessment, and ethics committees, high throughput drug screening, and predicting drug response for each patient, personalized medicine from tumor molecular features using Bayesian methods and neural networks. In 2060, machine learning, artificial intelligence, they'll design the molecules. And we've always been saying that statistical methods tell us when to terminate a clinical trial. These methods will tell us when to begin a randomized clinical trial, fill in information that we don't already know. Machine learning will write the protocol. AI will warn us of toxicity, regulatory hurdles, and unintended consequences of synthetic biology, and estimate the cost of development. Instead of a planned interim analysis, trials will terminate when cumulative posterior probability of an alternate hypothesis crosses a pre-planned threshold. So data are coming in from all of these different sources. And when some measure crosses a threshold, and the threshold might be called the coefficient of cogency. When enough information is available to determine that the alternate hypothesis can be supported. Very exciting. And this analysis takes all aggregate data into account, and the regulators and sponsors be following the same analyses in real time. They can be looking at dashboards like we look at dashboards for COVID-19 today, and see how close we are to approaching the coefficient of cogency. Jerry Cornfield, clinical trials are cogent description, he said, but he wouldn't recognize persuasive evidence today. Cornfield's cogent description was randomization and frequentist data analysis from a single trial. Is our idea of cogent description changing? Do regulators, insurers, physicians, and patients see cogent description differently? Must we all live with the regulator's idea of cogent description? Perhaps cogent description is not uniform. Different post-market analyses for different stakeholders, regulators, insurers, physicians, and patients. The New England Journal of Medicine, the JAMA, Lancet, the analyses we see there are the regulators' analysis, and it is the analysis that they find useful in their work. But other stakeholders might have other interests, different estimands, different priors, separate publication of results. Some journals, a separate journal will have an analysis that would be of interest to a physician group or of patients or insurers. Are we moving in that direction? Here is a table. I won't go through every cell here, but if you look in the first column, these are the different stakeholders and different objectives like risk-benefit and cost-effectiveness for the third-party payers. And analysis methods, regulatory is always frequentist, but perhaps we can use decision theory for patients and third-party payers, and quality-adjusted life years would be of interest to patients and third-party payers. Can this be analyzed after the drug is approved? My fourth hero is B.W. Brown, Stanford University. We've come full circle because he presented this paper at the ENAR statistical meeting, April 1979. And oddly enough, it was published in Controlled Clinical Trials in the same volume as my paper that I mentioned earlier. And in the opening paragraphs or opening sentences of his talk, he said, the analysis we do depends on the audience that we are approaching, that the audience that will read the analysis. Well, there must have been between 5 and 10 people who made a notation about that statement on their yellow pads, and probably they didn't pay attention to the rest of the paper. As soon as the Q&A came, all of these hands went up, and he was attacked. How can you say such a thing? Of course, there's only one analysis. Several post-market analyses, pharma regulatory issues. There's implications for a pharma company and having different analyses out there as far as the reception of their new drug, if it's approved. Regulatory and other analyses may come to different conclusions, but why not? Why shouldn't they? The FDA does not deal with the cost of drug, and regulators have no data on effectiveness, which we can get from this. More sophisticated safety analyses can come, aggregate analyses, risk-benefit, more models and simulation, and a role for real-world data in these analyses. Clinical trial methodology, I have always said, big R and little d. Big R because lots of research done, lots of dissertations, postdocs working on things, published in journals. Little d, little development. How many of them are ever used in an actual clinical trial? This is an opportunity for more methodology to be used. So what is common? 1980, 2020, 2060? The pursuit of cogent description. Randomization? Whenever cogent description is needed, some form of randomization is a best practice. Yesterday, today, and tomorrow. Thank you for listening, and now let the questions fall from the sky.